So, further questions? Am I in charge? Are we all back on my screen now? Okay, hopefully everyone sees my title slide, which looks a lot like everyone else's title slide except for the background, which is now black, because I'm fashionable. So the guys have sort of given you an overview of their individual facets of this larger group project, and I get to come along at the end and sort of sweep up the pieces. And what I intend to do, and I'll get you out to lunch fairly early, is sort of kind of give you a little bit more of the behind-the-scenes look at what works, what doesn't work, how we got to where we are, and hopefully by the end where we think this could go and be usable for the folks in this room. So again, this is a group project. This is a USDA NIMFAB collaboration that's been going on for a while. The names are shown here. You've heard about next-gen sequencing, so I'll spare you this. What I want to emphasize is this particular point. For next-generation sequencing as a diagnostic tool, and we're not the only people who have thought of using this, obviously. The power, of course, is the sheer volume of data that you get. The problem, of course, is the sheer volume of data that you get. And so the entire focus of this project has been looking for ways to cut down the data issue. Because the reality is, for pathogen and or strain identification, we don't need full genome sequences. When you do PCR, you're actually using 2 small pieces of DNA to identify something. The vast majority of that metagenome is really sort of superfluous and unnecessary, and it's just bogging the system down. So the goal here is to ignore all the irrelevant sequences, all your host sequence, all your identified sequences, and limit the bioinformatic processing of the rest of them, whatever else is of use. Getting scary here. Get closer and closer. So that's where EDNA came from, and I don't think it's actually been spelled out, but EDNA stands for ePro Diagnostic Nucleic Acid Analysis. Of course, EDNA was used because it's a catchy term that everyone knows in EDNA here and there. And basically this is just a bioinformatic tool designed to do exactly that, eliminate the difficulties working with large plant metagenomes. Now, typical metagenome analysis involves the following. You do a sample extraction, nucleic acids, whether it be RNA or DNA. You amplify that in some way if you need to. You put it through a next-generation sequencing platform. You then screen that. You assemble it, and then you blast it against GenBank or some subset, and you use a program like Megan to parse your data. EDNA seeks to do away with some of the problems in this system. Now, in typical metagenome analysis, the first steps are relatively straightforward. Assembly can be a little bit tricky, but we're getting better and better at assembling things, and the assembly programs today do a relatively reasonable job. The problem comes when you do the blast against GenBank or some subset of GenBank, because you are now dealing with 2 exponentially growing sets of data and doing a comparison. So your next generation sequencing run, your metagenome run, these are getting larger and larger as the technology gets better and better. In addition, you're taking these larger and larger data sets and then throwing them against GenBank, which is a non-curated, relatively immense pile of data that is also growing exponentially. So as time progresses, as technology progresses, and as GenBank gets larger, this process will get slower and slower. In addition, GenBank itself is inherently biased. Andres' slides were an excellent point for this. When we first started doing these analyses, we didn't have Megan hits coming back with all of these phytophthora, but as the phytophthora initiative took off, the phytophthora representation in GenBank became larger and larger. So if you do a traditional metagenome analysis for any eukaryotic sequence, you will now find that phytophthora exists in there simply because there's so many phytophthora sequences in GenBank. There's no phytophthora in the sample. There's just a huge representation in what you're calling your reference database. When you compare it to a biased reference database, you get biased answers. We needed to avoid that. EDNA does this, as the guys pointed out, by changing the pattern. So instead of querying the entirety of your next generation sequencing run against GenBank, we're going to develop query sets. And the program that they're using, which I don't know if that was spelled out either, is called Tools for Fingerprint Identification, or TOFI. And this is simply looking for diagnostic sequences that can either identify the species or a strain level. TOFI is the program we started with, but I don't necessarily think it's the end-all be-all. You could really use any sort of query development software. You could use Primer 3. You just have to change the setting. We've also sort of heard two different methods for developing these queries. When we originally started, we were using discrete query lengths, but as we moved on, we realized that that's actually not particularly necessary. It's more important to get good, clean, specific diagnostic sequences of any length than it is to say we need to keep them at 40 mer or 60 mer or 80 mer or anything like that. So this is something that may change as we move forward. I want to point out again that this is a work in progress, and we're adjusting on the fly as we go along. So in EDNA, you do the same thing. So you extract your nucleic acids, you generate DNA or cDNA libraries for your samples, you run your sequencing protocol. You then, instead of taking that entirety of the run, assembling it, and blasting it in a GenBank, you dump it as its own non-assembled sequence data. You create your own mini GenBank, and then you query that raw sequence with a series of your eProbes. And the guys have explained how that works. I want to spend a little time on this slide because it's the same schematic as the guys have shown, but I want to show you some of the steps that have been adjusted as we've gone through the process. So extraction to extraction. We tend to extract total nucleic acid. We started out with DNA extractions, but we rapidly realized that, well, that's going to be problematic for RNA viruses, for one. But two, looking at RNA gave us a lot more information. For one, we were then able to try and distinguish between a spore that had landed on a leaf and was just sitting there blank, or one that was actively undergoing an infection process. If it's producing RNA, it's actively undergoing an infection process. If it's sitting there as a spore, you're just going to get DNA. So RNA-seq has become a significant portion of this as well. All of that material then goes through an amplification step. We added in the whole transcriptome amplification, and then eventually we moved exclusively to whole transcriptome amplification because that also provides you whole genome amplification along the way. We started out using 454 sequencing. We're now working with Joanne Crouch's lab to try and transition this to Illumina as well as to other platforms. You develop your sequence samples database, and then you query it. If your pathogen is present, you can keep going by adding different eProbe sets to look for different things. If there's nothing present, you could stop right there. That's the basic principle of it. I know the folks in Canada have developed a metagenome amplicon-based system, and that you're going to hear about, and I encourage you all to attend the training on the 30th as well. We kind of feel like the two could be paired hand in hand if you simply treated this entire system a little bit differently. You do the same extractions, you do the same sort of collection here, but if you took this step here and added OTU amplification to it, you would then have a hybrid SSD that you could then query in multiple different ways. And this is something that hopefully Wen and I can talk about doing sometime in the future. Anyway, moving on. Here is what it looks like in text format. We'll just blow up to do that. What I want to get across is the point, though, that the power in this system is the ability to ask multiple questions just by changing your query set. Obviously, on a first pass, anybody in a diagnostic capacity wants to know on a species level what's there. So you build your first pass queries to do identification on taxonomic basis. But if you need more information, if you need strain typing, you can then go back and refine your answers by changing the query, changing the question. So instead of using a target that's this species, a near neighbor, which is the closest species, if you change that to where the target is your strain of interest and your near neighbors are other strains of the same species, you change your query set, you change the question you ask, and you can then start to do forensic analysis instead of just species identification. And of course, at Fort Detrick, they're all interested in biocrimes and things like that. So they were very keen on the ability to develop tests for things that have been genetically manipulated. So to do that, you simply develop queries based on common markers for genetic manipulation, antibiotic resistance genes, multiple cloning sites, things like that. So there, because you're not able to use full genomes, we do more of a tiling system where the entire antibiotic resistance gene is used as a set of eProbes. But you can just change the question and get a different answer. That makes this a very robust and flexible system. And obviously, people who are more clever than myself will be able to come up with even more interesting ways to address different questions. The nice thing about all this, of course, is that that same sample is stored as data. It's not stored in a freezer as DNA. If you go through the entire process of the queries and you don't find what you're looking for, later on when you find out about the next Liberobacter or the next disease that had no counterpart, you can go back and rebuild queries, query the same data, and retroactively figure out what's going on in something. And you can query it as many different ways as you can imagine. Of course, the keys to this are the selection of the proper query sequences, and for us, the minimization of that post-sequencing analysis. Yeah, I think I'll save that for just a second. So, how does it work? It's worked incredibly well and probably better than I would have expected when I first started this. This is some of Tony's data that he didn't show. Here, we're showing how you can identify both DNA and RNA viruses from the EDNA pipeline. On the top two lines, you're seeing 2 DNA viruses that were identified by probes, including a mixed infection. On the bottom, you see the ability to identify plumpox virus and RNA virus as a proof of concept. We're also able to strain type the plumpox in a sample using the same principle again. Just change your query set so that you now have M-specific or D-specific or EA-specific. You can now strain type your plumpox. That worked very well. All this is published in Pathology, by the way, if you're interested in it. So from a viral standpoint, we're able to detect both an RNA virus, a DNA virus. We're capable of detecting mixed infections. We're able to do strain typing. We were able to change the query set in the opposite direction. So by using general virus query sets, for example, potivirus query sets, we were able to identify a novel Tombus virus from a switchgrass sample as well. And that's a project that's ongoing, using the same sort of software to look for unknown knowns related to things that are there, but not particularly identified to a species level yet. Here's the cool thing for us. From a diagnostic standpoint, there aren't too many diagnostic labs that have their own computer clusters and the ability to do really heavy, in-depth analysis. We needed something that would work on a laptop and work reasonably reliably well. And so this is a comparison between the viral analysis on a laptop using EDNA, where it takes 14 seconds, to traditional Megan analysis on Oklahoma State's cluster, where it takes 9 hours. And that's a huge bonus. In bacteria, as John mentioned, we're capable of detecting multiple different types of bacteria. Whatever you develop the probe for set, it will detect. It can detect them in mixed infections. In addition, we were actually able to detect and distinguish between native Serratia marcescens and GFP-modified Serratia marcescens in planta, which is pleasing. We branched that out to look on human pathogens on plants. We had to take a slightly different approach because this is the one area where RNA sequencing doesn't work. There's just not enough RNA being produced in these cases. But none of these particular human pathogens are actually infecting the samples, but we can still find them just being present. So we were able to detect E. coli 157 on bean sprouts. We can detect Salmonella on bean sprouts. And we're working on detecting Listeria as well. So this has capacity to detect human pathogens that are simply residing on the surface of plants and food tissues as well. As Andres mentioned, this is capable of detecting both oomycetes in Phytophthora and Pythiums, as well as true fungi, Puccinia and Phacoryza. He has done some preliminary strain typing with Pythiums, but that's definitely a work in process as well. This is particularly pleasing because I thought if we were going to have a problem, this is where it was going to occur. There's a lot of conservation amongst eukaryotic genomes. Part of the reason Andres had to spell out and develop an additional novel system for kind of separating what was a true positive and not false positive is because of the interplay between eukaryotic hosts and the eukaryotic pathogen genomes. there's enough conservation there that we have to worry about more than, well, if you just throw random sequences of a certain GC content at it, will it matter? In this particular case, we had to both do decoy probes, but also look at whether or not we were generating false positive matches. And that's part of the process that he published, or this paper is actually in press now. In our lab, we've adapted this to do what we call insect EDNA. In particular, we developed probes for the P aphid based on a mitochondrial genome. We are then able to, in a metagenome, detect P aphids. Probe subs have also been developed for leafhoppers because those are also useful. And what we anticipate this would be sort of a way to look for vectors. Eventually, the goal here is to be able to take an insect trap from any location. Instead of having to key out morphologically all the insects within the trap, you could simply grind them all up, extract nucleic acids, and use EDNA to detect both the vectors and potentially the pathogens from mixed traps. And this actually, to me, has more immediate use than perhaps using this as a true diagnostic immediately. So bottom line, it's done everything we need it to do. It detects RNA viruses, DNA viruses, detects bacteria, detects oomycetes and fungi. It can detect vectors. It's useful in pathogen discovery, and we're incredibly pleased with how well it's going. Which isn't to say we're quite done with it yet. There's still some refinement that needs to happen, but at least it's got the potential to do a lot of the things we need it to do. The folks in regulatory circles are very excited about it because it addresses some of the problems they have. They know that next generation sequencing is a real possible tool for them, but we're reluctant to jump into it because of the infrastructure required to handle the data. So EDNA does the kinds of things that it needs to do in terms of trimming data down to a usable size. We don't have to worry about the size of GenBank. We don't have to worry about the ever-expanding size of next-generation sequencing runs. This could work with any platform. It could work with any organism right out of the box. For them, it's hugely valuable because now there's a potential for a single protocol that could detect any pathogen they have to worry about in any host that they have to worry about or any matrix they have to worry about, including soil or water or air. And for those of you who aren't familiar how regulatory labs work, at least for ISCO certification, every technician has to be certified on every assay that they do, and they have to be certified regularly. So for APHIS's point of view, this is a potential scenario where you wouldn't have to teach a technician every single assay for every single pathogen on every single host that they're going to encounter. You could teach them one protocol. They could be certified on it, regularly certified on it, and that would hold them through for a good long time. EDNA also has the advantage of being a don't ask, don't tell diagnostic. And what I mean by that is if you do a metagenome analysis, as you see when you looked at Andres's Megan run, you get back a lot of information, a ton of information. And from a diagnostic standpoint, that's not really necessarily useful, particularly if it's based in a biased system. And our system, the GenBank, is biased toward pathogens. So it gives you a lot of calls. Oh, there's a lot of Phytophthora infestans in there. Really, there's not. But that still is a legally actionable thing. What APHIS needs is, what regulatory agencies need is a system wherein they can ask for, is this pathogen present and only this pathogen present? Because I don't want to have to go back to the farmer and tell them, no, you don't have plumpox, but you have 6 other podoviruses, Phytophthora, Pythiums, as well as some Vaccinia. They would prefer to have just the assay for the pathogen that they're interested in, and this particular system does that for them. In advantages, it should work with any NGS platform. Again, this is something that we're in the process of getting ready to test. It's got versatile simultaneous detection of all taxa of plant pathogens, everything from RNA viruses right on up to eukaryotes. It's flexible in terms of specificity. If you need to look for an unknown Pythium, if you need to look for an unknown Pseudomonad, you change the way the queries are built. You can look for things that are related to but not exactly the same as what you know about. You can change that probe set to ask any number of questions. You can look for genetic modifications. You can look for human pathogens on food. You can detect vectors, and it's useful in pathogen discovery. These are all valuable things. But there's certainly some things that are limiting to us at this point in time. There's definitely limited detection, as probably the questions brought out. We need a certain pathogen read abundance to be able to detect anything, and it's somewhere between that magic window of 0.5% and 5%. This is something I feel will improve, one, because sequencing technology is improved. As you change platforms, Illumina provides you a lot more reads. PacBio provides you a ton more sequence information than the 454 juniors that we started with. When we started with 454, it was simply because that was the only available platform at the time that had long enough reads to do what we needed to do. Now they all have what we need. So improving sequencing technologies will definitely improve our limited detection. I feel like we have not demonstrated this yet, but if we worked on ways to improve probe representation, we would increase our limits or improve our limits of detection. That is to say, the more probe representation you have from a particular genome, the more likely you are to find one of the sequences within a mixed metagenome. And that's just sort of a mathematical equation that you can talk about if you're interested. But I also feel like we could work with the existing system that's being developed in Canada, add in OTUs, and improve the limited detections for bacteria and fungi, which is where we're really going to have the problem anyway. Viruses get to very high levels. I don't have too much of a worry about ever detecting viruses, but I do have worries about finding fungi and bacteria. Currently, there's a time limitation for eProbe development. The actual EDNA analysis doesn't take very long. Developing eProbes, particularly when you're talking about eukaryotic systems, takes a while. So eventually I would envision this being a web-based service, and I'll cover that a little bit more in a bit. Lastly, the major limiting factor is the lack of available information for your targets. So for viruses, it works very well because there are plenty of viral sequences. For bacteria, there's getting to be enough, so it's a very usable system. But you saw the sort of tricks that Anders had to go through to try and find enough genetic information to even build eProbes. For soybean rust, for Saccharisa, he could not get a full genome, so he has to work exclusively with ESTs. ESTs are wonderful, they provide a lot of information, but it's also the most conserved part of the genome that's most likely to be conserved between Saccharisa and other fungi, Saccharisa and the host for that matter. So in those particular cases, the lack of genetic information was limiting. This is especially true for the insect situation. There's not a lot of genomes out there. This is why we're working strictly with mitochondrial genomes at this point. Again, this is something that as time goes on, scientists do more and more research in genomics, this will improve. And the last disadvantage is we really, as you folks are probably aware from having tried to load this and run this a little bit on your own, we really need to develop this into a user interface, perhaps even a Windows-based user interface that will be readily usable and understandable without having to know how to do command line. So these are all things we feel we can fix, and this is how we're going to try and do it. Oops, there's my fail on the corner. The other thing, sort of the elephant in the room, how much does it cost? Obviously, we know next generation sequencing is not trivial, but the reality is in certain situations it might already be feasible from an economic standpoint. If you look at the cost of a single 454 junior run, for us it's about $1,500. That gives you 140,000 reads. If you need 1,000 reads to be able to identify your pathogen, you really need about 10%, 10,000 reads. You could, in theory, run somewhere between 7 and 14 samples per run. If you probe that with 15 different sets, that allows you to do 210 tests per run, and you divide the total cost by the number of tests. That gives you something that's more reasonable. It actually comes down to about $7 per test. Now, realistically, if you're only interested in looking for plum pox, this is not the economically feasible reason to do this. But if you're actually running a germplasm certification program where you do have to do 15 tests, now this becomes something that's reasonable, particularly when you start to factor in labor time. In our vision, we'd like to see this implemented to the big data USDA initiative. We'd like to divide EDNA into two packages. We'd like to divide it into an eProbe maker. Hopefully this would be something that would be web-based and housed by the USDA on the big data initiative, maybe through iPlant. This would be something where a user goes in, brings their target, finds their near neighbor, asks the web-based service to build the eProbes for them, and they would be sent back to them. And then the user would download the EDNA portion of it. They do the diagnostics on their own laptop. It would be a little bit like your classic GenBake scenario. So the eProbe maker would allow you to either develop, put in accession numbers or the actual FASTA files for what you're interested in developing probes for. You could select specificity parameters, and then you'd get an email back with your FASTA eProbes. And of course, users would be allowed to develop their own eProbes as well. But the point of having this nice eProbe maker would allow you to develop a consolidated database of eProbes, so that every time we did not have to reinvent the wheel. As users register for this, it would be sort of incumbent that they would provide the eProbe sequences as public information for future users as well. The EDNA portion of it would, of course, run on your own laptop. It wouldn't look all that differently. You would enter your metagenome FASTA file. You select from a drop-down menu or uploaded probe sets. Again, set your parameters and then let it run. And you can, again, query that data as many different ways as you feel like. I really want to acknowledge the people behind this because it's most definitely not me. John, Andres, and Tony did the bulk of the work developing the system. They have it up and running, and they're the ones who successfully tested on so many different things. Ruchi took this in a different direction and looked at machine learning as a way to try and find unknowns. Trina is the postdoc who took on the foodborne pathogen approach, and now she's faculty at Oklahoma State as well. Diana, Aaron, and Andy have all contributed out of my lab. Diana and Andy in particular have been working on both viral EDNA and insect EDNA and pushing things into new platforms. And of course, the PIs also helped out quite a bit, but everybody in white sort of kind of did a little bit less. That's about it. So that's the theory behind EDNA. That's where we're headed with it, and that's where we see it potentially interplaying between the two groups that are on the phone today. I'd be happy to take any questions. Also take at this point comments, suggestions, but bear in mind you have 20 minutes before you go to lunch. Thanks.